How about your experience last week? We didn't have class. We're off, right? I hope all of you are safe and always take care of your health. Today, I will share with you an interesting topic. It is enzyme. Slide, please. Enzyme is a biocatalyst. Biocatalyst is a catalyst that can increase the rate of reaction that occur in a living system. Let us see at the figure below. Molecule A as a starting material will react to form product D through several reaction steps catalyzed by a several enzyme. Then, what is enzyme method? Let us see this one. Next, please. All enzymes are protein. Is it true or false? George, can you choose one of the two? Safira, how about you? Yes, false. It is the answer. Because in reality, almost all enzymes are made of highly specialized protein. Ribozymes are a molecule with catalytic properties. And the same thing, upzymes are antibodies with catalytic properties. Now let's move to activation energy. In chemistry, activation energy is a term defined as energy that must be overcome for a chemical reaction to occur. The minimum energy required to start a chemical reaction. The activation energy of a reaction is usually defined or denoted by energy activation and given in unit of kilojoule per mole. Let us see the graphic on the right side. The horizontal one is a progress of the reaction and the vertical one is a free energy. Let's see the black line. The black line course of reaction without enzyme, and the red line is a course of reaction with enzyme. If we look at the energy activation, then energy activation by using enzyme should be shorter than without enzyme. Then in this case, there isn't any relation to the delta G. Sometimes enzyme need to be tanu. Cellular need and condition vary from cell to cell and change within individual cell over time. sometimes it is necessary to inhibit an enzyme to reduce a reaction rate. And this is more than one way for this inhibition to occur. Let's see in the figure an example of regulation in a blood clotting. We should discuss from the left side first, the image of blood vessel. The image of this blood vessel will release the clotting factor. Then the image of this blood vessel will be repaired by platelet aggregation. In this case, we should see in this formation of a platelet plaque. Then from this platelet plaque will develop of clot. Then let us see in the upper side. In the upper side, there is a clotting factor. A clotting factor will be affected conversion of prothrombin into thrombin. And this thrombin will affect the conversion of fibrinogen to fibrin. This fibrin is a kind of protein that is insoluble. Then that's why in discussing of this blood clotting, we can see that this should be happen from the make blood vessel into the development of clot. So this clot will repair the image of the vessel. Next. Now let us discuss about a mode of metabolic regulation. Enzyme regulation in metabolism can be as a allosteric regulation, covalent modification, proteolytic activation, feedback regulation, isoenzyme, compartmentalization, and metabolic specialization of organ. Let's see one by one. Allosteric regulation. Using a reversible non-covalent binding of regulatory molecule to the enzyme. Allosteric enzyme have additional conformation induced by a modulator. Let's see the figure in this slide. Allosteric inhibition. We should determine first this enzyme one. This enzyme and one allosteric site can be attached by inhibitor. After attaching of this inhibitor to the enzyme, then the active site will be, the conformation of this active site will be changes, then the substrate could not attach to the active site. Then in that case, the reaction is blocked. But let's see in the allosteric activation. In the allosteric activation, let's see. In this allosteric activation, this activator will attach to the enzyme 2. After attachment this activator, then this active site will be open to catch up the substrate. Then the reaction can be occurred. Next slide, please. Now let us move to the feedback inhibition. Feedback inhibition is a specific type of allosteric enzymatic activity regulation. Enzyme is specifically inhibited by the end product of the pathway whenever the concentration of the end product exceeds to the cell requirement. Let's see, for example, in this slide. We'll determine first from the upper side. Threonine. Threonine will attach to the active site of the enzyme. Then, for several method, this threonine will convert it to isoleucine. This is the end product of the enzymatic reaction. But anyhow, this end product will attach to the binding site of the allosteric of the enzyme. So now then the active site of this enzyme has already changes again, then it will be block the substrate that has been used at the same time in the previous explanation. Next slide, please. Now let us discuss about mechanism of inhibition. This mechanism occurs for a reversible inhibition reaction. Reversible inhibitors are divided into three groups based on their binding behavior. Let's see, for a sample, the first one is competitive inhibitor. In this case, we can see that inhibitor compete with the substrate in the same active site. The second one, non-competitive inhibition, we can see that the reason not the only inhibitor compete with the substrate, but in this case we can see one of the inhibition is in the active site, so there is compete of substrate with inhibitor. But also after changing this reaction enzyme plus substrate into enzyme substrate complex, then this enzyme substrate complex also compete with the enzyme inhibitor plus substrate. Then the other one is competitive inhibitor. This the only inhibit the enzyme substrate complex. So from the mechanism, we can see that the three totally different one each other. Now let us see the case study methanol poisoning. Methanol is a poison, antifreeze, pain, thinner. Not because of what it does to the body itself, but because enzyme at the edge oxidize it to formaldehyde, which is potent poison. A treatment of methanol poisoning is to give to the patient ethanol. Why is this effective? Let's see. Methanol is poison, but it's not because the methanol is safe, but because a product of the conversion of methanol into formaldehyde. This formaldehyde is completely poison. So in this case, in the neutralization of this poisoning, we should give to the patient ethanol. Like ethanol is metabolized by NADH, but enzyme affinity for ethanol is 10 to 20 times higher than it is for methanol. Now, let us see what happened with this poison methanol. Ethanol is a competitive inhibitor of methanol to alcohol dehydrogenase. As ethanol is added, less methanol can bind to alcohol dehydrogenase active site. Formaldehyde is produced at slower rate, so the patient doesn't get sick. So now you can see that you have methanol. Methanol is poison, but it's not because the methanol is safe, but it's because of the product of methanol, formaldehyde. This formaldehyde is completely poison. So to treat this one, we have to give to the patient ethanol. So there is a competitive inhibition of the reaction. Yes, let us see the other. Covalent post-translational modification. As you can see, for nowadays, more than 500 different molecules are known to be modified using this method. Common reversible covalent modification of enzyme, phosphorylation, adenylation, ADP ribosylation, and methylation. A donor molecule provides a functional moiety that modify the property of the enzyme. Let us see the two figure in this slide. This is the upper side. The upper side is an inactive enzyme. So in this case, there is a phosphorylation by using ATP. So inactive enzyme will convert to active enzyme after phosphorylation by catalyzing with enzyme kinase. In the downside, we can see that inactive enzyme has already activated. Sorry to say, has already phosphor in it. To activate this inactivated enzyme, it should be converted by hydrolyzing this phosphor group. Now we can see that the active site do not need the phosphate group in the enzyme. Now let us see proteolytic activation. Some enzymes are secreted in inactive form called proenzyme or zymogen. Zymogen is inactive because it contains additional polypeptide chain that block the enzyme active site. Activation of zymogen occurs with proteolytic process. Now let's see this figure. Let's see this zymogen surrounded by environmental factor. The active site of this zymogen has already blocked because of a polypeptide chain in this case. Then this polypeptide chain in zymogen will be autocatalyzed activated, and this zymogen will be open. Then the catalytic activity can be occur again. That's say like this activation by hydrochloride, pepsinogen into pepsin, autoactivation, pepsinogen, pepsin, activation by other enzyme, let's say like trypsinogen into trypsin and prothrombin into a thrombin. Now let us move to the feedback regulation. You will not to be confused with feedback inhibition because there is a bit the same feedback and feedback inhibition and feedback regulation, a bit the same, but it is not the same, completely different. Feedback regulation means that the end product in the reaction decrease the rate of enzyme synthesis at the level of gene expression. Let's see this figure. A synthesis of cholesterol from acetyl-CoA. By going for several steps, then acetyl-CoA enzyme A will produce cholesterol. From this pathway, at this we can determine there are three probably the main thing molecule that we should remember. The first one is a linosterol, a cholesterol, and oxysterol. Linosterol and oxysterol will affect the synthesis of HMG-CoA reductase. So these two molecules will affect the synthesis of this enzyme. So that's why, as I have already mentioned before, there's this case related to the level of gene expression. Next slide, please. Now let us discuss about isoenzyme. Isoenzyme are enzymes that differ in amino acid sequence yet catalyze the same reaction. They display different kinetic parameters such as Cmax, or different regulatory properties. For example, of this isoenzyme, we can see this reaction. Regulation of LDA in a cell during heart attack. Let's see lactic acid, pyruvic acid. In this case, we can see that lactate dehydrogenase consists of two types, as you can see in this figure. H4 expressed in the cardiac muscle, M4 expressed in skeletal muscle. Partial block of blood flow as a result of heart attack release H4 isoenzyme to bloodstream. In this case, used as a marker. So in this discussing isoenzyme is completely interesting to understand, and I think it's completely also interesting to do the research related to this isoenzyme. Next slide, please. Now let us discuss about control of enzyme level. The amount of enzyme as well as their activities are controlled. The rate of synthesis and degradation of many regulatory enzymes are altered by hormone. Hormones are chemical messenger of human body. Let's see this body, human body. From the brain, we have a brain, we have several organs in the stomach. Then every organ, every gland will produce hormone, and this hormone will need by several methods in using their hormone activity to control the enzyme level. You can see from this figure, let's say this one, thyroid, hypothalamus, and kidney and ovary. Okay, this organ will produce hormone. Then these hormone are chemical messenger to the human body. Next. Now let's move to hormone in metabolism regulation. Let's see this one. This is also very interesting. Cortisol is made by adrenal gland. Cortisol increase in response to stress. Many cortisol receptors are located on our cell. An example of this one is cortisol can induce gluconeogenesis and increase blood sugar, while prevent sugar intake from extracellular system. Let's see the figure in this slide. Cortisol will up. It's mean the concentration of the cortisol up. But what's the effect of this cortisol? It's so many things to understand and so many things to explain about this cortisol. But from this figure, you can see that this is related to, let's say, the fat will be increased, brain food intake, liver glucose generation, muscle glucose uptake will be decreased. So in discussing this cortisol, you should remember that this cortisol is very important in discussing the metabolism regulation of hormone. I mean, hormone regulation in the metabolism pathway. Next slide, please. Now let's move to the compartmentalization. Compartmentalization of metabolism pathway permit integration and regulation of metabolism. The cell is compartmentalized and cellular structure play a part in bringing order to the metabolic pathway. It makes the reaction more accurate, targeted, and less interfering with each other. Let's see, this is a cell, right? This is a cell, this is a cytosol. And in the inner side of the cell, there is a mitochondria. Either cortisol and mitochondria completely different in case of discussing the metabolism pathway that occur in the cytosol and mitochondria. In cytosol, let's say like glycolysis, pentose phosphate, and fatty acid synthesis occur in cytosol. But in mitochondria, there are many metabolic pathway that occur. For example, citric acid cycle, oxidative phosphorylation, ketone body, and so on. So in this, we should discuss interplay of both compartments. So it means that there is a compartmentalization of the reaction in this case. Okay, next slide, please. Each enzyme are located on different compartments, as I have already mentioned before, thus enabling the compartmentalization of the metabolism reaction. Let's see this one again, the same thing like as I have already mentioned before. This is the table related to, let's say, this is cytoplasm, mitochondria, Golgi, and many other. We call this one as a compartment. Compartment. In each compartment, in each compartment, there is or there are many things that probably one each other different in a metabolism pathway. Next slide. Now let's move to metabolic specialization of organ. Regulation in eukaryote is enhanced by the existence of organ with different metabolic rule. In this case, we discuss eukaryote. If we discuss eukaryote, we should understand the difference between eukaryote and prokaryote. Eukaryote, for example, human is a karyote cell. Bacteria probably a prokaryote. Metabolic specialization is the result of differential gene expression. The same thing, there is a related one that I have already mentioned before. Each organ, each gland has their own metabolic specialization and will, let's say, will produce different metabolic pathway in each organ. Next slide, please. I think that's all for today. Thank you so much. If you need more explanation about this one, I'll happy to answer the question. And thank you so much. Good afternoon. Take care of yourself. See you again next week. Bye-bye. Thank you.